Pollen storage is our today's topic, which is in continuation with pollen viability, which we learned in the last chapter or last topic in the chapter Palynology. Now, as far as pollen storage is concerned, it is nothing but simply storing viable pollens with intact or intact vigor in appropriate containers like glass or plastic vials for an extended period of time. Viable pollens are those pollens who have the ability to germinate. So obviously, beforehand, you will check the viability of pollen grains of a particular taxa, and based on that, you would like to store these pollen grains for extended period. That is, the naturally we have already grouped the pollen grains into categories like long-lived pollen grains, which are viable for 6 months to 1 year. Say, for example, from family Fabaceae, Pinaceae, Anacardiaceae, Vitaceae. So all these members, they already have the viability almost up to 1 year. And if you want to have these pollen grains for more than that period, then obviously you would like to go for pollen storage. But there are some pollen grains like from the family Gramineae, Cyperaceae or Commelinaceae. So these pollen grains are short-lived pollen grains because they remain viable only for a few minutes or maximum up to couple of days. So these kind of pollen grains you can store and you can extend their period of viability. And that is what the storage is all about. And for that you need to have these appropriate kind of containers of glass or plastic vials, and then you have to store the pollen grains with appropriate methods. So in this topic we are going to study about what are those appropriate methods. Now such containers which are stored, they are stored in the desiccators with a dehydrating agents. Why? Basically to control the humidity, because humidity is one of the important factors which we have mentioned earlier. Temperature and humidity, these are the two factors which are controlling the pollen viability. So basically in order to control the same, we are putting all these containers in the desiccator with a dehydrating agents. We can also use the saturated solutions of different salts in order to obtain the required humidity. I'm sure you have heard about Lycopodium. It is one of the pteridophyte member, and the spores of this Lycopodium are used as a diluent. Diluent is nothing but, see, already the number of pollen grains which you are using for storage are limited, though that quantity is very less. So in order to have more number, you are adding Lycopodium spores as a diluent before the storage to increase the bulk of the pollen and prevent the wastage of pollen sample during this artificial pollination. But obviously there are some points you must remember before you are using any of the plant as a diluent. Lycopodium is very commonly utilized. You can also use talcum powder, you can use corn or wheat flour also. But then there are certain points you must consider, and that is the diluent must be non-sticky, non-hydrating, and keeps the viability of the pollen grains high. Okay, these are the basic, you can say, criteria you must utilize in order to choose the diluent. They must be non-sticky, non-hydrating, and they are keeping the viability of pollens high. So if you are done with your basic study of these criteria, you can use any particular diluent. So like Lycopodium spores are definitely one of the most sought after spores to be utilized for the process of pollen storage. Now, there are two basic methods or types of pollen storage. One is called as short-term pollen storage, and the second, but obvious, is a long-term pollen storage. Obviously, it is depending upon the time span you're looking for. I told you last time one of the basic uh, use of stored pollen grain is to be utilized in plant breeding experiments. In the plant breeding, so there are two types of pollen storage. One is short-term pollen storage, and the other one obviously is a long-term pollen storage. And both of these methods are different on the basis of the time span you're looking out for storage of the pollen grains. While discussing pollen viability, I made a point that the pollen grains which are viable, you can collect them and you can store them, especially for horticulture or agriculture purpose, because both of these kind of plants can be utilized and can be, uh, with the help of plant breeding you can make the better varieties. So in the plant breeding experiments you require male parent, the male donors for the pollen grains. So obviously at that time these pollen grains can be utilized. So based upon like certain plants, their pollen grains are long lived and some of them are short lived. So based on that you can choose the method which is not only appropriate for the particular plant, but at the same time it must be economically viable. Okay, because some of the methods are comparatively very expensive. You must have the access to those particular instruments, and that is the reason why you have to select a particular method based on your goal as well as whatever is available to you. So the first one is short-term pollen storage. As the name suggests, it is a method where you are storing the pollen grains for comparatively short period of time. Now we know that in general, low temperature and relative humidity are favorable for most of the taxa. However, pollen of a large number of taxa can be successfully stored for a limited period of time through the manipulation of storage temperature and humidity. Pollen grains can be suitably stored in a glass or a polythene vials or in appropriate containers. Now, in case of unsealed containers, suitable dehydrating agents like silica gel or various concentration of sulfuric acids or saturated solution of different salts are used to maintain the required relative humidity. So temperature and humidity, these are the two important factors, and you can literally manipulate with both of them in order to get the desired effect. Now, especially for the storage purpose, you can also utilize the lycopodium powder, that is lycopodium spores. You can use talc powder, you can use corn or wheat flour. They can be utilized as a diluent. And we have already mentioned the factors you have to consider while choosing the particular diluent. We can also talk about pollen storage in organic solvent. Now, there were two scientists, Iwanami and Nakamura, both two Japanese scientists in 1972, first demonstrated the use of different organic solvents for pollen storage. Now, they includes the very common benzene, petroleum or diethyl ether, acetone, chloroform, etc. Now, their efficiency varies greatly for different plant species. So literally it can be the research work wherein you have to choose several groups of plants, either genera or species level, and then you make a concentration of different organic solvents. So you can imagine it's a very, very huge work, but then people have done it, and accordingly they are suggesting for a particular plant species which particular organic solvent in which particular concentration is suitable. Pollen grains stored in non-polar organic solvents like benzene or diethyl ether and cyclohexane, they retained viability and showed very little leaching of phospholipids, sugars, and amino acids into the solvent. Now, what is leaching? Leaching is nothing but to give out. Leaching is, we are also using this word for soil. When the water falls, or rather rain falls, the inorganic salts which are present in the soil, they leach out, they goes to the bottom. Okay. Now here, the pollen grains, they have the membranes. Okay. Now in those membranes, there are phospholipids, there are sugars present within the pollen grain, and obviously it is a great source of amino acids. So when you are putting the pollen grains in the organic solvents for comparatively a short or long period of time, there is a possibility of leaching. So you have to also find out in which particular concentration the leaching is more and in which concentration leaching is less, because viability, maintaining viability is one point, but at the same time without any loss to the contents of the pollen grain. On the other hand, extensive leaching of substance and loss of viability is seen in polar organic solvents. So we can conclude over here that non-polar organic solvents are comparatively better than those of polar organic solvents. Citrus pollen maintained viability in different organic solvents for 3 months. Maize and many insect pollinated species, it must be wind pollinated, not insect. Maize is a grass. So maize alone is a wind pollinated, and the other insect pollinated species, they show the storage in the suitable organic solvent at 4 degree centigrade for 35 to 40 days. Now you remember maize is a grass family, and if you remember, in grass family, we mentioned that the pollen grains are short-lived pollen grains, which are only for few minutes to couple of days. So if maize is a member of gramineae, whose normally pollen grains remain viable for a couple of days. Now you can see here, when I'm using organic solvent, suitable organic solvent at 4 degrees centigrade, the pollen remain there for almost 35 to 40 days. So almost you're extending the storage period from 1 day to the 40 days. And that is a huge success in some of the plants. Then we are also looking for the pollen grains to retain their viability for a very long period of time. So that is referred to as long-term pollen storage. Now storage, especially at temperature below 0 degrees centigrade, usually slow down the metabolic activity of a pollen, resulting in gradual decrease and finally total loss of pollen viability. One of the important factor for losing pollen viability, we have mentioned and that is loss of respiratory substrate, that is glucose. Because if the metabolic rate is very high, along with high relative content of the water, due to very high rate of respiration, the respiratory fuel, that is glucose, would be finished off, and as a result the pollen remain non-viable. When you store when you store any food material in the refrigerator, you're not maintaining 0 degrees centigrade, but you can. So if you are putting the plant material, food material in the refrigerator, your uh, basic intention is to preserve at low temperature, because at low temperature the chances of multiplication of microorganisms are very less. At the same time, the basic metabolic rate of those plant or food products from within would be comparatively very low. So spoilage is delayed. I'm not saying there is no spoilage. Spoilage would take place. अगर फ्रिज में आप कोई चीज 2 महीने के लिए ऐसे ही रख दो, then obviously it is going to spoil. So what I mean to say is it is a delayed. So same thing with pollen grain also. Take the pollen grains and store them at 0 or maybe less than 0 temperature. And obviously to maintain that temperature, you require specific kind of instruments. So this method is not economical method. I told you earlier that you have to decide upon the method depending upon the material you are using, number one, number two, the time span you are looking for, and number three, economically viable. If the procedure requires lakhs of rupees and you don't have fundings, obviously you're not going for it. You'll purchase some of the non-polar chemicals and you'll try to store them because expensive. The chemicals would be comparatively less expensive than those of the other materials. But if your lab is already equipped with the very good refrigerators and some of the other instruments which we are looking for, then obviously you'll go for the long-term storage, pollen storage. So we are talking about storage at sub-zero temperature, sub-zero, less than 0 degrees centigrade. Using a storage temperature of -10 degrees centigrade and -34 degrees centigrade, the longevity of bicellular pollen, that is two-celled pollen. One cell is vegetative cell, second is generative cell. And the pollen with original low content of moisture has been successfully extended between 1 and 3 years. 1 and 3 years, almost up to 3 years, you can increase the pollen storage. So the first method we have is that of freeze or vacuum drying, which is lyophilization, also known as lyophilization. Pollen of different taxa, especially those who are tolerant to desiccation, can be successfully preserved for a long period of time by this particular method. Now, this method involves rapid freezing of pollen to sub-zero temperature of 60 degrees centigrade. It must be -60 or -80 degrees centigrade using inert gas like helium or nitrogen, and then gradual removal of water under vacuum sublimation. So here is a technology, and obviously the technology comes with a cost. But yes, this is one of the wonderful technology where you can see you can store the pollen grain for a comparatively longer period of time. In this particular vacuum drying, the pollens are directly exposed to a vacuum and then simultaneously cooling. In number of taxa, when freeze drying is combined with lyophilization, then storage and viability of pollen have been found to be very effective. So it's a combined effort of both freeze drying along with vacuum drying. That vacuum drying is called as lyophilization. So if both are successfully utilized, they are comparatively more effective. But then we have one more better method than that of freezing or vacuum drying, and that is cryopreservation. See, this is one of the basic principle of cryopreservation. In cryopreservation, before discussing cryopreservation, let me discuss this particular diagram where you can see a cell at 37 degrees centigrade, that is the optimum temperature. And then we are subjecting the cell slowly to less than 0 degrees centigrade. Okay. Now there are two possibilities when you are cooling the cell slowly, correct? Then there is a possibility of formation of ice. Okay. And
Yes,
there is a possibility of injury of that particular cell. But when I'm doing this particular cooling rapidly, still there is a possibility of formation of ice injury. Okay. So there is still possibility of the cell to be dead. But then what I'm doing here you can make out the chances are comparatively lesser with rapid cooling rather than that of slow cooling. So in case of vacuum drying or freezing, what you are doing is rapid freezing of pollen to sub-zero temperature. I'm not saying that rapid cooling will not have the injury into the cell, but then it is all about taking a chance. Out of 100 pollen grains, there is a possibility that few pollen grains would be dead. But then for remaining, say 70-80% of pollen grains who survives, it is the best method available. Okay. But when you are doing slow cooling, the chances of having the dead cells would be comparatively much more. And that's the reason why rapid cooling is preferred over that of slow cooling. Yeah. Then we are talking about cryopreservation. Here you can make out some gas is coming out. It is nothing but nitrogen gas. And nitrogen gas is having its temperature minus 196 degrees centigrade. And that is the reason why liquid nitrogen is used. Liquid nitrogen, whose temperature is minus 196 degrees centigrade, is used for cryopreservation. It's a long-term preservation, which can be also be done by ultra low temperature, which is also called as ultra low temperature, ranging between minus 70 to minus 196 degrees centigrade. A reduction in the pollen water content below a threshold level before low temperature exposure seems to be important for achieving viability. Thus, partially dehydrated pollen possesses less freezable water and can survive deep freezing. Partially dehydrated pollens are used for this particular process so that they can survive for deep freezing. We are aware of graminaceous flowers. The graminaceous pollen grains, they are the one who are very short-lived pollen grains because they are having comparatively high amount of water, almost 35 to 60 degrees, 35 to 60% of water when they are liberated from the anther. Thus, immediate freezing would cause irreversible structural damage as a result of ice formation. And therefore, the water content of pollens needs to be reduced before cryostorage, which is, however, problematic as there is a rapid loss of viability with a decreasing water content. Water loss without any detrimental effect in pollen viability ranges between 50 to 80%. This, however, again depends on the species and its genotype. So when you are deciding upon which method is a suitable method for your pollen grains, you have to consider all these factors, that what kind of pollen grains are they, what is the relative humidity, percentage of water content, what is your purpose of storage of pollen grains, and then based on all that you have to decide upon which method is comparatively suitable. And the result is here. You can see here we can successfully use cryo storage for pollen grains for number of different species. Beta vulgaris storage in cryopreservation, you can store it up to 1 year. Capsicum 16 months. Carica papaya 485, more than 1 year. Glycine max is nothing but soybean for 21 days. Helianthus anus, that is sunflower, for 4 years. And Solanum tuberosum, that is potato, for almost 2 years. That is how you can storage those of pollen grains. Coming to what is significance of pollen storage. Now we have seen there are two basic methods, short-term and long-term pollen storage. You have to decide which method is suitable for your pollen grains. But then question arises, why I am interested in storing the pollen grains? The systematic research in pollen storage started at the end of 19th century. And now we have today large number of crop species, including vegetable species, fibers, fruit crops, forage. The forage is leafy vegetables, cereals. For all these, we have got pollen storage strategies is desirable. Why? Because we are trying for better and better varieties. And when you are trying for better and better varieties, one of the best suitable method is plant breeding. And in plant breeding, you require to collect the pollen grains, store the pollen grains, and utilize as per your requirements. So obviously pollen storage is desirable. Second important significance is genetic conservation through pollen storage. I'm sure you have heard about seed banks. So seed banks are nothing but the way you can store the genotypes. When I'm talking about a pollen grain is haploid. It is having one set of chromosome, and that is something called a genome. So for the storage of this genome, you can always conserve the pollen grains. So genetic conservation through pollen storage is desirable for a variety of horticulture, agriculture, plant species, since pollen is known to transmit important genetically heritable characters. Pollen cryobank. Pollen is a product of genetic recombination and can provide a reliable source of nuclear genetic diversity at the haploid stage. I'm sure in tissue culture you must have heard about production of haploid, androgenic haploid plants. So how they are produced? You have to take pollen grain, immature pollen grain, as a explant, as a starting material for tissue culture, and then based upon that you have to make the based upon that you will get the so-called callus, and then you treat it with the hormones and you develop into the plantlets. That is how it goes. All those plants which are produced by such methods are called as androgenic haploid plants. So although genetic conservation through pollen storage does not accomplish the whole genome conservation, a plant breeder involved in genetic enhancement of a given horticultural or agricultural crop could have access to the facility which is called as pollen cryobank. And from this where he can draw pollen parents of his choice in the process of breeding a new cultivar, a new variety. So suppose I am into developing new plant varieties, and I know that there is something called as a pollen cryobank from where I can obtain these particular pollen grains so that I can carry out the plant breeding experiments in my fields. So such cryobanks are there, very much there. You must have heard about IARI, Indian Agricultural Research Institute. So who is into agricultural research? So they have set up such cryobanks all over their head offices. What, as a scientist or as a horticulturist or as a agriculture scientist, what I need to do is I have to approach these particular offices and I have to apply for obtaining specific type of pollen grains through them. So these pollen grains can further be utilized as a starting material, cross with those of the female parents, and you have to obtain specific type of F1 hybrids, and those F1 hybrids you can further uh, select and utilize as per your requirements. So such cryobanks, pollen cryobanks, are nothing but the laboratories where there is a facility of cryopreservation, which is nothing but preservation of pollen grains at minus 196 degree centigrade. Okay, so these are the different significances of pollen storage.